Hello, is it audible? Hello, it is audible. వాళ్ళకి మా ఇవ్వాలి లింక్ ఇవ్వాలి ఇదే లింక్ లింక్ ఏ లింక్ Okay. And with regard to this, the factors proved to be the genetic material, it is the DNA proved as the genetic material at least for the majority of the organism. So, as the word itself conveying at least for the majority of the organism means in some organisms, in some viruses, the few viruses, RNA act as variety genetic material. That is a key point. In only few viruses, RNA act as genetic material. RNA mainly act as messenger RNA, carries message, genetic message from the DNA. Why not DNA not involved directly involved in case of what it is the exhibition of the character? DNA is a long molecule and DNA it is contained in the nucleus. For synthesis of the protein, amino acids are required. Amino acids are which are available in case of cytoplasm. That's why it triggers its duty to what it is the mRNA. Now mRNA takes the charge to translate what it is genetic message to form what it is the polypeptide. That's why it is information passed on to the mRNA as a cassette information in case of mRNA. So mRNA mainly act as what it is messenger RNA. mRNA also act as adapter RNA. Otherwise it is called soluble RNA. That is, adapter RNA means amino acid attached to what it is, the tRNA. And also act as rRNA, that is ribosomal RNA. The rRNA mainly it is a structural RNA. And rRNA also act as what it is, catalytic RNA. The catalytic RNA in case of the prokaryote, the bacteria, it is 23S. And case of eukaryote, it is 28S. 28S, ribozyme. So, enzyme is always a protein. This concept is going to be wrong. Most of the enzymes which are proteins, that means there are some enzymes which are non-protein, nothing but what it is, ribozyme, that is 23S rRNA and 28S rRNA. And most of the enzymes formed by translation. Some of the enzymes formed by transcription. So, and the DNA and RNA, the polymer that is made up of what it is many monomers form what it is the polymer. In a polymer, in each monomer, it differs in whether nitrogen base or sugar or phosphate. If you take the DNA, it is a polymer of what it is the nucleotide. In every nucleotide, what it differs with regard to what it is nitrogen base. The sugar and phosphate is the same. So, each monomer in a polymer differs in nitrogen base of the sugar, but not in case of what it is the phosphate in one nucleic acid. In both nucleic acid, it differs in case of the nitrogen base as well as what it is the sugar. Nitrogen base in case of the DNA, it is the thymine. In case of RNA, it is going to be what it is, uracil. And in this, how DNA is the genetic material. So, formation of mRNA from the DNA and genetic code, and all in case of what it is very first paragraph information. DNA, it is an interesting genetic material. Why it is abundant and interesting genetic material? The haploid content of the DNA is the genome, and computer-based analysis of the genome heralded branch, it is the genomics. In case of very first paragraph information. And in case of the molecular biology language, 1 kbp is equal to 1000 base pairs. In case of first one, pi x 174 phase. It is single stranded DNA. Pi x 174 phase. And in second one, Coli lambda phase 48502 base pairs, and it is linear double stranded DNA. Linear double stranded. Linear, it is single stranded. And third one, it is E. coli, that is circular DNA. 4.6 into 10 to the power of 6 base pairs. And case of the human DNA, it is linear double stranded DNA, 3.3 into 10 to the power of 9 base pair in case of haploid content. So it is key point, linear single strand, linear double strand, circular and it is double strand, linear it is double strand. So in case of 5386, so 5386 nucleotides, in terms of the pairs means by 2 it gives what it is nucleotide pairs. It is nucleotides. Nucleotides. Number of phosphodiester bond in double stranded DNA molecule is equal to total number of nitrogen bases minus 2. In case of single stranded DNA, number of phosphodiester bond is equal to total number of nitrogen bases minus 1. Number of phosphodiester bond in case of the double stranded DNA molecule is equal to number of phosphodiester bond into 2 plus 2 in case of double strand DNA. In case of single stranded DNA, number of phosphodiester bond is equal to number of phosphodiester bond is equal to n minus 1. Phosphoester is equal to phosphodiester bond into 2 plus 1. So in case of single strand, into 2 plus 1 of the phosphodiester bond gives what it is number of phosphoester bond. In case of double stranded DNA, number of phosphodiester bond into 2 plus 2, that is going to give what it is phosphoester bond. So it is nucleotide, it is base pair, nucleotide base pair, it is nucleotide base pair, it is nucleotide. In diploid content, 6.6 into 10 to the power of 9 base pair. In haploid content of the DNA, how many glycosidic bonds? 2 into 3.3 into 10 to the power of 9 base pair. In diploid content of the DNA, in case of the human DNA, number of glycosidic bond 2 into 6.6 into 10 to the power of 9 base pair. So we have to extract the gist of what of information, the length of the DNA based on the number of what it is base pair. In molecular biology language, 1 kbp is equal to 1000 base pair. With regard to what it is, the second paragraph information. And with regard to this nucleotide, polynucleotide, So the DNA, it is having the components, it is the sugar, phosphate, nitrogen base. The sugar, it is the pentose sugar. C5H10O4 in case of deoxyribose sugar. C5H10O5 in case of the ribose sugar. So ribose sugar, deoxyribose sugar differs with which carbon it is? Second carbon. There is a deficit of what it is? 1 oxygen. That's why 2-deoxyribose. And in case of the phosphate, H3PO4 minus. So oxygens carry much negative charge. That's why DNA it is negatively charged. So biologically it is the phosphate. And nitrogen bases which are purine nitrogen base and pyrimidine nitrogen base. Purine and pyrimidine nitrogen base. So largest nitrogen bases which are purine nitrogen bases. The purine nitrogen bases which are adenine, guanine. Pyrimidine nitrogen bases thymine, cytosine, uracil in case of nucleic acid. So total number of nitrogen bases in all nucleic acid is equal to nothing but what it is 5. In DNA nothing but what it is the 4. In RNA nothing but what it is the 4. And adenine, it is a dicyclic compound. Guanine is a dicyclic compound. And it is having carbon nitrogen rings. It is called heterocyclic compound. So all dicyclic compounds are heterocyclic. Thymine, cytosine, uracil, it is a monocyclic single carbon nitrogen ring. All monocyclic compounds which are heterocyclic. So all nitrogen bases which are heterocyclic carbon nitrogen rings. Total number of carbon nitrogen rings in all types of the nitrogen bases 2 plus 2, 4, 5, 6, 7. Number of carbon nitrogen ring in all types of the nitrogen bases in nucleic acid it is the 7. What is the ratio of the carbon nitrogen rings of the purine and pyrimidine? 2 plus 2, 4 and it is the 3. So 4 is to 3. So common pyrimidine nitrogen base it is cytosine. Common pyrimidine nitrogen base going to be bonds with what it is guanine. So we have to check whether common pyrimidine nitrogen base or common pyrimidine nitrogen base bonds with. Common pyrimidine nitrogen base is cytosine. That is going to bond with what it is the guanine. So adenine it is the 6 amino purine. Guanine 2 amino 6 oxy purine. Thymine 2,4-dioxy 5-methyl, that is thymine. And uracil 2,4-dioxy, it lacks for it is the methyl group. And in case of cytosine 2-oxy-4-amino. And you have to check in this which nitrogen base having this amino group. Which nitrogen base having what it is oxygens. that forms for it is one type of for it is the question with regard to the nitrogen bases. And bonding in case of for it is the nucleic acid. And nitrogen base attached to first carbon sugar by N-glycosidic linkage. 9,1 glycosidic linkage in case of for it is purine. 1,1 in case of for it is pyrimidine. In case of purine, it is attached with the 9th atom of the nitrogen with the first carbon sugar. That's why 9,1 glycosidic linkage in case of purine. 1,1 glycosidic linkage in case of the pyrimidine. And it is at a phosphate attached to the 5th carbon by means of what it is phosphoester bond. It is one dehydration, it is a mononucleotide. In mononucleotide, how many bonds are present? Glycosidic bond, phosphoester bond. There is another nucleotide. The 5th carbon, the phosphate it is attached. It is one nucleotide. It is one nucleotide. It is the 3rd carbon. The phosphate it is attached to what it is 3rd carbon sugar. It is one phosphoester. It is one phosphoester. So 3,5 phosphodiester linkages. In adjacent dinucleotide, type of the bond, glycosidic bond, phosphoester bond, phosphodiester bond. In a dinucleotide, it is a single strand. In a dinucleotide, number of phosphodiester bond is equal to what it is? Only it is 1. So, number of nitrogen bases 2. Number of phosphodiester bond it is 1. Number of phosphoester bonds it is 1, 2, 3. So, total phosphoester bond it is 3. In a dinucleotide, number of nitrogen bases 2, number of sugars 2, number of phosphates 2, number of phosphodiester bond 1, number of phosphoester bond it is 3. So 3,5 phosphodiester linkages. Don't get confused with synthesis of the DNA, 5-3 direction. Synthesis of RNA, 5-3 direction. Translation in 5-3 direction. And phosphodiester linkage formation in it is 3,5 phosphodiester linkage of what it is one strand. So it is the dinucleotide. And opposite nucleotide hydrogen bond. So to check the question, adjacent nucleotides are held by phosphodiester bond. Opposite nucleotides are held by means of what it is the hydrogen bond. And the two strands of the DNA exhibit what it is the polarity. So two strands of the DNA which are asymmetric. In these two strands, the 5' and 3, 5, 3. At 3 OH 5 phosphate, 3 OH 5 phosphate. So 3' and 5' it is anti-parallel and it is opposite direction. So what type of what it is the bonding in case of what it is the nucleic acid. And deciphering the structure of what it is nucleic acid, derivation of what it is nucleic acid. Nucleic acid discovered by Frederick Miescher in nucleus of what it is pus cells. Nucleic acid named by Altman. And structure of the DNA discovery it is attributable to what it is Watson and Crick. Even though it was attributable to Watson and Crick, they are successful based on what it is experimental evidence of the previous scientist. Nucleic acid literature because of the contribution of the previous scientist. That is Wilkin, Franklin, Khargov, new scientist Pauling. According to Wilkin and Franklin, X-ray diffraction, previously it is called X-ray crystallography, to assess the structure of macromolecule, a sort of helix, according to Wilkin and Franklin. And Khargov, purines and pyrimidines exist in what it is 1:2 1 ratio. A plus G is equal to T plus C. So A plus G is equal to T plus C. It is the purines pairs with varieties, the pyrimidines. The purines pairs with varieties, the pyrimidines is applicable to varieties, the double stranded varieties DNA. Not it is a single stranded DNA. And the GC ratio is more than what it is 80 ratio in case of what it is prokaryote. 80 ratio more than GC ratio in case of what it is eukaryotes. That's why in case of the prokaryotes, the DNA denaturation, it is very slow. Denaturation in case of eukaryote, it is very fast. There are more GC pairs than what it is 80 pairs in case of prokaryote. So, GC and AT pairs varies from what it is organism, prokaryote and eukaryote. But A plus G is equal to T plus C, that is universal in case of the double-stranded DNA molecule. And Pauling, according to Pauling, hydrogen bonds are established between what it is nitrogen bases. So nucleic acid literature available and experimental evidence available based on the contribution of Wilkin, Franklin, Korgoff and Pauling. Watson contributed for double helical model of what it is the DNA. So the double helical model of the DNA attributable to Watson and Crick. So in this hallmark of excellence of the contribution of Watson and Crick is complementarity. That complementarity is also based on what it is observation of what it is Korgoff. That is also key point. Complementarity hallmark of excellence of the discovery of the Watson and Crick is complementarity. Whenever purine comes, oppositely pyrimidine. Whenever pyrimidine is oppositely what it is, the purine. So, based on what it is, the Korgoff analysis. That is one point. And deciphering the structure of the DNA, elucidating the structure of the DNA was elusive for number of years. It was ignored for number of years because technical limitation in extracting a long intact polymer. Why it is a long intact polymer? It is a long molecule and combination with RNA, protein, carbohydrates, lipids. So many number of things are there. So it should be extracted from those things. So there are technical limitation in extracting a long intact polymer. That's why elucidating the structure of the DNA was elusive for number of years. That way it is dormant, kept what it is unnoticed. That means not properly deciphered. And in this, the salient features of the DNA, length of the DNA it is variable. We don't know this length. Length of one spiral it is 34 angstrom. And 10 angstroms is equal to 1 nanometer. So 34 angstroms means 3.4 nanometer. So pitch of the DNA, length of one spiral it is 34 angstrom, 3.4 nanometer. 34 angstrom the DNA and these two spirals which are, it looks like a twisted ladder. There is exact distance between the two strands of the DNA. That is going to be wrong. Approximately, it is the similar, that is the 20 angstrom. The distance between the two strands of the DNA is approximate, that is 20 angstrom. So, what is the diameter of the DNA in 10 spiral? 10 spirals or 15 spirals or 20 spirals, the diameter is going to be 20 angstrom. Why diameter is going to be 20 angstrom? That is constant throughout the length of the DNA. Purine pairs with the pyrimidine, pyrimidine pairs with the purine, vice versa. That's why diameter it is constant. Length of one spiral 34 angstrom. 34 angstrom can accommodate what it is 10 pairs of nucleotide. So far number of DNAs which are discovered, more than 12 DNAs which are discovered. There is no universal geometry for all the DNAs, but there is one DNA that is a balanced DNA with a definite hereditary stability. What do you call it is BDNA. So we are discussing about what it is the BDNA, that is the balanced DNA. So that is the balanced DNA. It is the BDNA that is having definite hereditary stability. And the base pairs stack one above the other. The three key points. The one it is base pairs stack one above the other. And even though hydrogen bonds which are weak, they provide stability because of many hydrogen bonds. And presence of what it is thymine. Stability to DNA. And this point you come across in case of this RNA world. So thymine provide the stability to the DNA. And the 2 points which are base pairs stack one above the other. And presence of hydrogen bonds many, even though they are weak, there are many hydrogen bonds are present that provide what it is stability. That is one point. And genetic implications with regard to the structure of DNA became so clear after proposition of what it is replication of the DNA. How it got cleared? What are the intricacies involved? According to Mendel, the progenies must be similar to that of what it is the parent. Here, the two DNA strands, the one DNA molecule is going to give what it is the two DNA molecules which are exactly similar to what it is the parent. Why? In one DNA molecule, two strands act as what it is template. As the two strands act as what it is template, the two DNA molecules which are formed. So the word it is genetic implications with regard to the structure of the DNA became so clear only after proposition of replication of DNA. How? The two daughter DNAs resulted by replication, which is exactly similar to the parent DNA. Molecular weight of the two daughter DNA, it is exactly similar to that of already the parent. Two strand molecular weight is not same, but molecular weight of each DNA molecule similar to that of already the parent strand. That is also one of the key point. Why two daughter DNA molecules are similar exactly to the two DNA molecule? Because in DNA, two strands act as what it is template. That's why it is exactly similar to what it is the parent DNA. So genetic implications became what it is so clear. And as the DNA transfers information to what it is mRNA, so DNA to mRNA, mRNA to what it is the proteins. DNA to DNA, it is auto catalysis. This maintain what it is genetic integrity. DNA to mRNA, it is transcription. mRNA to protein, it is translation. Protein, exhibition of character. So DNA responsible for autocatalysis as well as what it is heterocatalysis. DNA responsible for regulating the metabolism through heterocatalysis by synthesizing what it is the protein. DNA responsible for maintaining genetic integrity through autocatalysis. So DNA to DNA autocatalysis, DNA to mRNA transcription, mRNA to proteinity translation. Total it is heterocatalysis. That is central dogma of molecular biology according to Crick. Just simply it is the Crick. So maybe question A, B, C like this. So it is replication, transcription, translation, heterocatalysis, autocatalysis, genetic integrity, and regulation of metabolism. The regulation of what it is metabolism. It is heterocatalysis. And according to Crick, the flow of information, it is unidirectional, but it was differed by Temin and Baltimore. That is reverse transcription. On RNA, DNA formed. What do you call it is reverse transcription? RNA dependent DNA polymerase, what do you call reverse transcription? Discovered by Temin, it is Teminism. The word it is Teminism. Discovered by Temin and Baltimore in retrovirus. RNA dependent DNA polymerase. And DNA dependent RNA polymerase, it is transcription. So here remember Temin, Baltimore, reverse transcription. The phenomena it is Teminism. And in calculating this length of the DNA, distance between adjacent base pairs, because 1 nanometer is equal to 10 to the power of minus 9 meters. So distance between adjacent nucleotide, that is 0.34 nanometers into 10 to the power of minus 9. It should be multiplied with what it is the total number of what it is nitrogen bases. In case of the mammalian cell, 6.6 into 10 to the power of 9 base pairs. That is going to give what it is 2.2 meter long DNA. That is accommodated in case of the small nucleus, 10 to the power of minus 6 meters long DNA. That is very small. That is nucleus. How the long DNA is accommodated by means of packaging and otherwise what do you call compaction. So long DNA molecule accommodated in a short chromosome that is contained in a very small nucleus, that is possible by means of packaging and compaction. For this packaging and what it is the compaction, in case of what it is prokaryote, eukaryote. In prokaryote, real chromatin organization it is absent. Circular, almost circular, naked, double-stranded DNA. In case of eukaryote, that is the DNA, it is contained in case of what it is the chromosome. And in case of the prokaryote, even though nuclear membrane is absent, DNA is not scattered. Why DNA is not scattered? DNA held with what it is non-histone protein has large lobes within what it is nucleoid. So genetic material of the bacteria, what do you call it is nucleoid. That is circular, naked, double-stranded DNA molecule, almost circular, without chromatin organization. Such a genetic material in case of bacteria, that is called genophore. And in case of eukaryote, it is called chromosome. Organized genetic material in eukaryote is considered as chromosome. Unorganized genetic material in case of the prokaryote considered as nucleoid or incipient nucleus, or it is genophore. Double-stranded DNA, that is naked. That is naked means it is without what it is histone protein. And it is having non-histone protein as a large loops in a nucleoid. And the packaging is so simple in case of prokaryote. And packaging in case of the eukaryote is highly complex because it occurs at several levels. It requires what it is histone as well as non-histone protein. So, histone protein it is as DNA negatively charged, histones which are positively charged. In this, lysine and arginine. So to link with the genetic code also. Lysine and arginine which are positively charged, side chain having the positive charge. That's why the histone, the protein, it is what it is positively charged. And non-histone chromosomal protein, NHC, non-histone chromosomal protein. The histone proteins are required at initial level of packaging. Non-histone chromosomal proteins are required at higher level of the packaging at the time of formation of what it is chromosome, like a DNA polymerases, RNA polymerases. So, which are non-histone chromosomal protein. And in case of histone, the histone, it forms an octamer. In this H2A, H2B, H3, H4, it is an octamer. So, number of types of histone present in case of octamer, 1, 2, 3, 4 types of what it is histone. And the DNA wrapped how many turns? 2 turns. 2 turns means should not be taken as what it is 34 plus 34. The 2 turns of the DNA having 200 base pairs. So each nucleosome, it is having 200 base pairs of what it is DNA. So 2 turns of the DNA should not be treated it as what it is 34 plus 34. 2 turns of the DNA having 200 base pair. And this octamer, above this it is having H1. As the 2 turns of the DNA around the histone octamer, that forms what it is nucleosome. This nucleosome appears as beads on a string of chromatin that present in nucleus in electron microscopic view. And this H1 protein sees the entry and exit of what it is, that is which protein remarkably useful in the formation of the chromatin fiber, that is H1 protein. Which protein is going to seal the entry and exit of the DNA? It is H1 protein. Which protein do not occur as a pair? That is H1 protein. Extra nucleosomal protein, it is H1 protein. In between to linker DNA, what is present? H1 protein. Number of types of histones involved in case of nucleosome, 1, 2, 3, 4, 5. 5 types of histones which are involved. Number of types of histones involved in case of the core, 4 types of histone. How many H2 types are there? It is 2 types, H2A, H2B. How many H2 histones are there? 4. How many types of H1s are there? 1 type. How many H1s are there? It is 1. How many types of H4s are there? 1 type. How many H4s are there? 2 H4s are there. Like this, you can extract the question from this. So, one nucleosome that forms what it is 200 base pair. In case of mammalian cell, 6.6 into 10 to the power of 9 base pairs. So for 200 base pairs, 1 nucleosome. For 6.6 10 to the power of 9 base pairs, how many nucleosomes? So we have to calculate that like this in case of the mammalian. So no nucleosomic concept in case of what it is the prokaryote, that is held as what it is larger loops. So packaging of what it is with regard to the DNA. And there is a word heterochromatin. And euchromatin. For this it is S-caramine. It is a stain. And chromatin loosely arranged, it takes less time and it is genetically active, transcriptionally active, it is euchromatin. And chromatin, it is tightly packed, which takes much strain, dark in color, genetically inactive, that is transcriptionally inactive. What do you call it? Is heterochromatin. Why differential staining occurs? Because of even and uneven coiling. Such a differential staining because of even and uneven coiling. What do you call it? Is heteropyknosis. The three words: euchromatin, heterochromatin. The stain, it is histocaramine, and it is heteropyknosis. Cell drinking is nothing but what it is? Pycnosis. As differential staining because of what it is even and uneven coiling. That's why what do you call it? Is heteropyknosis. Okay, it is. And in case of the next heading, search for genetic material, it extract the case of search for genetic material. So it reached to molecular level, which because the factors measure the nuclein and Sutton and Boveri, what it is the chromosome. So what is the molecular basis of what it is inheritance? Because the chromosome having the DNA, RNA as well as the protein, which is the biochemical responsible for what it is inheritance of character. That is the first experiment by Griffith, transformation in case of what it is that is Streptococcus pneumoniae. And the pneumonia, Streptococcus pneumoniae, is a chain of what it is the round shaped bacteria in this two strain, S strain and R strain. S strain is going to have what it is the mucus coat, and R strain it is without having what it is mucus coat. And this S strain, as it is having a polycoat, it is virulent, and R strain it is avirulent and it forms the rough colony. S strain forms the shiny colony. When S strain got injected into the mice, it dies. When R strain injected into the mice, it alive. When heat killed S strain injected into the mice, it alive. During heat killing, what are supposed to be undergone denaturation? Protein, RNA, DNA undergo denaturation. If you see the protein, RNA, DNA. At high temperature, the protein, the tertiary structure of the protein is going to be disrupted. Disulfide linkages, hydrogen bonding is going to be disrupted. Denaturation. And 2OH present at what it is, the second carbon is going to be labile and degradable. So RNA is going to be degradable. It is not going to be genetic material. And DNA, the two strands unwinded. The heat killed S strain The mice, it is alive after injection. Heat killed a strain live or were introduced, it dies. Because R strain transformed into what it is, a strain. Suppose they took strains, 10 A strains. They later found that A strain to R strains. That means which got transformed? R strain got what it is transformed into what it is. Transformed by it is a strain. So R strain got transformed into what it is, the S strain. And the transforming principle the unable to be deciphered by the Griffith. Avery, MacLeod, McCarthy, biochemical nature of the transforming principle discovered. That biochemical nature of the transforming principle, Avery, MacLeod, McCarthy, they used DNA ases, RNA ases, proteases. Proteases, they used it digests what it is the proteins, transformation unaffected. RNA ases, it digests what it is RNA into nucleotides, so it is transformation unaffected. DNA ases, transformation is going to be affected. What is the difference between DNA ases and DNA? DNA is a polynucleotide. DNA ase is a polypeptide. So DNA ase act on what it is, polynucleotide. DNA polynucleotide. DNA ase polypeptide. So DNA was the genetic material, but it is not unequivocally accepted, not universally accepted. Chemical nature of what it is discovered by Avery, MacLeod, McCarthy. Before Avery, MacLeod, McCarthy discovery, they thought that protein was responsible as a transforming principle, the genetic material. According to Avery, MacLeod, McCarthy, it is DNA, but it was not universally accepted. Universal acceptance of, that is, the DNA was the genetic material, Hershey and Chase. What do you call the experiment? It is side by side experiment. What do you call the experiment? It is the transduction experiment. Virus, that is mediated gene transfer, is nothing but what it is, transduction. So I have to link it with what it is, 3-point: transformation, conjugation, transduction. Transformation, Griffith, focus pneumoniae. Conjugation, E. coli by Lederberg and Tatum. Transduction, Ginder. So here transduction by Hershey and Chase to prove DNA was genetic material. So earlier protein versus DNA versus RNA, 3 are there. Protein, DNA, RNA. Protein versus DNA versus what it is, RNA. Now the protein versus DNA versus RNA got reduced to what it is, DNA versus the protein through his experiment, transduction, side by side experiment. Who used P32, S35 radioisotopes. And in these steps it is labeling, infection, blending, labeling, infection, blending, and what it is, centrifugation. So virus is initially labeled with what it is, P32, S35. During this labeling process, the virus, it is having a coat, the protein coat, and inside which it is the DNA. As a protein, it is having what it is, sulfur. So protein labeled with what it is, the sulfur. Phosphorus labeled into what it is, the DNA. as a protein, that's S35 labeled into the protein because the cysteine and methionine having variety sulfur. So dash and dash amino acid having sulfur, cysteine and methionine. That's why S35 labeled into variety the protein. And P32 labeled into the DNA, phosphorus present in case of variety is the DNA. And so labeling it is over. And labeled viruses made to infect with what it is the bacterial cells. What do you call it is infection. So bacteriophages attached to the bacterial cell with the help of what it is tail fibers, otherwise what do you call prongs at specific receptor site. So specific receptor site present on bacterial cell wall. Organs of attachment are tail fibers. Tail fibers otherwise called what do you call it is prongs. Okay. And after this infection, bacterial cell treat viral genetic material as its own genetic material and makes what it is viral particle. Once viral DNA enters into the bacterial cell, it takes control over total metabolic system of the bacterial cell. That's why viruses which are obligate intracellular parasite. For metabolic system, it depends upon what it is, the host. It makes what it is viral particle. So from bacterial cell, the coat proteins, it is separated by blending. The coat protein still attached to the bacterial cell separated by means of what it is the blending in a blender. And inside this bacterial cells, what particles which are made? Viral particles which are made. So in one, inside this P32 and outside this S35. In supernatant, S35 present. In one experiment, protein did not make entry into what it is this cell. In one, that is the P32 enters into what it is the cells. So DNA enters into what it is the cell. So DNA proved as a genetic material, unequivocal proof of the genetic material. Suppose they used N15. So it is not possible to discover that whether the protein or otherwise it is DNA, because the N15 labeled into the protein as well as what it is the DNA. That is also the key point. So labeling, infection, blending, centrifugation. In supernatant, what they observed? S35. Inside the bacterial cells, what they observed? Viral particle. Viral particles separated from the bacterial cells by a process called centrifugation. Coat proteins from the bacterial cells separated by means of what it is blending in a blender. So labeling, action, and blending and centrifugation in this very key point. Universal acceptance, local proof that DNA was a genetic material. DNA versus protein versus RNA versus was reduced to DNA versus what it is the protein. Transduction experiment, side by side experiment. Who used P32, S35. What are the steps? What is supernatant? Inside the cells to separate the viruses, what is the process? Centrifugation. To separate the coat protein, what do you call it is the blending. So in this key point, unequivocal proof, Hershey and Chase. DNA was the genetic material. Okay, this session it is over. So we'll see the topic in case of the next session. Some hindrances in this session. Okay, we'll cover in case of the next session.